So I'm going to talk today largely about what we're doing in Singapore to try to understand um, how we can intervene in humans to affect human aging and how we can use animal models to best tackle that problem. And I've used this slide for a long time, so I won't spend much time on it, but I just want to give a very brief introduction in case anybody's not thinking about aging in the perspective that most of us do now. And that's that, we've spent a lot of the research, and still in that research community, it's like this, but it's very focused on individual diseases. Therapies are very focused on individual diseases. You get diagnosed with something and you get treated for it. But we know these things have commonalities. And we also know, probably you already know this if you're listening to this talk, that aging is probably the biggest commonality for all of these chronic diseases. Matt Kaberlein and I were talking recently. We've been using this slide on the right for like 16, 17 years now. So I guess all that means is that we're getting old. But, I think that it's finally starting to resonate in a broad community that maybe we should actually tackle the common cause of everything in addition to just focusing on individual diseases. And many people recently have suggested that I've been going around saying aging is the biggest medical challenge, biomedical challenge of this century. And then COVID happened and everybody started disagreeing. And I just wanted to make the point that the biggest risk factor for mortality due to COVID-19 is being over the age of 70. And that's the biggest risk factor independent of chronic conditions or chronic diseases. So there's something about aging that also affects your resilience when you get an infection. And that shouldn't have been very surprising because the same is true for influenza here on the right and many other acute diseases. And so what we should have been doing all these years is using this slide on the right, which is that aging drives chronic diseases. It also drives at least the response to acute infectious disease. And it, of course, drives functional decline as well. I'm not going to show data on that today, but we all know probably from firsthand experience that as you get older, you can't do some things as well anymore. So it's even bigger than the slide on the left. I think the importance of tackling aging has many, many downstream benefits. And this is our fundamental approach in Singapore. The common healthcare system works like on the top where you're healthy for a while. We don't do much for you. Probably you don't go to the doctor's office regularly. We don't do much testing. We say, get some exercise and don't really do much prevention. And then at some point in your life, you get sick, you start getting a disease, maybe it's diabetes or some other chronic disease. Then you get multiple diseases. And as you get multiple diseases, it becomes harder and harder to treat them. And then eventually it's too much and you die. And all we're saying is let's do our interventions in the green zone and try to extend health span, keep the period of time when you're disease-free and highly functional. And the interventions that do this in animal models not only extend health span, but they extend lifespan as well. Usually health span goes up more than lifespan, at least with the one I'm going to show you today. And so we're actually achieving compressed morbidity, which I think is a big goal in human intervention studies. So I'll tell you about our clinical approach at the end, but it's really geared toward this concept. Now, as you know, there are two papers that came out 10 years ago now, the Hallmarks of Aging and the Pillars of Aging. And actually, both groups are now doing new versions of this. So look for that hopefully soon. Right now, it's sitting in my Word file. I'm not doing anything, but I got to get working on it. And the idea is to understand the mechanisms of aging. This has been something that's been going on for a long time. And the only point I want to make, and I've made this point several times, is that I don't think it's so much the individual pillars of aging that's important, although we can tell stories about all of these and how they impact aging. It's the interconnectivity. So I look at healthy aging as maintenance of a homeostatic network that keeps you healthy in the face of changes that are happening with aging. And that could be damage, it could be infection, it could be all kinds of things. But your body is resilient, it responds, and this network brings you back to some sort of equilibrium. And it's only when enough things happen to the network that it starts to fall apart, that you can't respond anymore. And that's when you really get these long-term chronic diseases. So most of the interventions that I'm going to talk about, I think really what they do is preserve the function of this network. And of course, it's fine to talk about intervening in aging, but it only is relevant if there are interventions that work or that can be tested. And now there's a wide array of these. There are lifestyle interventions in the dark red circle. I'm not going to talk much about those today, although we do work on some of them. Drugs and small molecules in the sort of medium circle. And that's where I'm focusing most of my work. And then more radical interventions like gene therapy, stem cell therapy, reprogramming that are rapidly coming on board, but maybe not quite ready for human testing. We do some work on stem cells and cell replacement therapy. I'm not going to talk about today. But the amazing thing is that if you look over here on the right, this is from the Drug Age database recently. There are now over 1,000 distinct drugs that have been reported to have some effect on aging in some species. And actually, over 110 of them are linked to aging in mice. And most of our studies have been in mice. And so what we're trying to do is to take a reasonably agnostic approach to go through and find small molecules that impact health span and lifespan in mice, validate those in mice, and then take those into human studies. And so everything over here on the left are things that we're actively working on, a few other things as well that are not on the list. Happy to talk later about them if you want to contact me. I'm going to focus today on alpha-ketoglutarate and Gemfibrozil, but we have very interesting data on urolithin. I think this is in our animal models working. Done a lot of work on rapamycin. We published a recent paper on spermidine, and we also have unpublished data on vitamin A. And so I think that a lot of interventions are likely to work in the right context in mice, and we're trying to understand how they relate to each other and begin to combine them together. So these are the two drugs I'm going to talk about today, Gemfibrozil and alpha-ketoglutarate. Gemfibrozil is in an earlier stage. Alpha-ketoglutarate, we have some human data already. And so that'll give you kind of a spectrum of how we're thinking. Gemfibrozil is not widely known in the longevity field, but it's a widely used drug. It was identified as and used under the trade name Lopid, used by Park Davis, and they should use it for hypertriglyceridemia, hypercholesterolemia. Often it's used in people that don't respond well to statins. It's a fibrate, and I'll talk about that in a minute. This is the key data that got it clinically approved, or part of the key data, which showed that it lowered triglycerides in the blood and raised HDL cholesterol. So this drug has been used for a long time. It's a very successful drug. And the mechanism of action is the drug is reported to be that the drug comes into the cell. It binds to this complex, which contains a transcription factor, PPAR alpha, disrupts the repressor component from this complex, allows the transcription factor to go in the nucleus and transcribe a whole set of genes that are involved in getting rid of lipids, cholesterol efflux, HDL production, better storage of lipid droplets, etc. So we started out thinking this might be a mechanism that underlying the aging effect. So this is work done by Chong He, who's now working at a company in the US, and Charmaine, who's a graduate student who's getting close to finishing her PhD in my lab at the US. And so why did we get interested in Gemfibrozil? Well, it strangely goes back to yeast. We had found that Gemfibrozil extends lifespan in yeast and in worms. And we wanted to know what it was doing. So the first thing we did is we knocked out the gene that looks like PPAR alpha in yeast. Excuse me. And that had no effect. Gemfibrozil still extended lifespan. So it didn't seem like that's what was going on. And so then we're looking for new mechanisms. One of the things you can do in yeast is that you can used this growth array where you have 100 different growth conditions for yeast, and you see which ones are sensitive to adding the drug. And a lot of these different growth conditions involve different carbon sources. And what we found is Gemfibrozil doesn't affect the growth of the yeast unless you put them on dipeptides and tripeptides as carbon sources. So, yeast and humans and mice have mechanisms to take up dipeptides and tripeptides. They have receptors that do that, and they can use, in yeast, you can use it for energy production very easily. And when we put yeast on these different media sources with dipeptides and tripeptides, Gemfibrozil blocked the growth. So that led us to think that maybe we're inhibiting amino acid uptake. And if you just looked in cells on the bottom here, sorry, I'm not using the laser pointer. I think by now I can learn to do that. So if you just look at amino acids in the cells, you see that most of the amino acid levels go down in cells when you expose them to Gemfibrozil. And this is an elegant little yeast experiment. I'm not doing much yeast now, so I kind of miss it. So I'm going to force you all to pay attention to this one. What you do is you take a dipeptide that has a toxic amino acid that's non-native, and when you put it on this little piece of paper and let it diffuse into the media, it kills yeast because this dipeptide gets into the yeast and this toxic amino acid kills it. And what we were able to show is that when you add Gemfibrozil, the yeast grow because they don't anymore. So we had the mechanism by which lifespan was being extended. And so we went back and remember I told you that Gemfibrozil extends lifespan in worms too. So we went back and looked at worms. And if you add this is just a fluorescent dipeptide now, not toxic. But we can see where it goes in the worm, and you can see it gets into the intestine and it goes into the intestinal cells, or the cells lining the intestine. But when you add Gemfibrozil, the dipeptide stays in the intestine; it doesn't go into the cells. So the same mechanisms seem to be happening in worms. And I'm not going to talk about this, but we could see the same effect in human PC3 cells as well. And in fact, when we looked in human cells and looked at amino acid levels in the presence of Gemfibrozil, we got almost the same result in yeast, a reduction in most amino acids. So we wanted to know what the target of Gemfibrozil was, and we decided to do a screen. So we screened about 6,000 proteins using this SETSA analysis. And essentially what you do is you raise the temperature of the extract, expose it to the drug, and you use the premise that when a drug binds to a protein, it usually stabilizes it. So if you have a drug binding to its target, that target will still be folded normally at higher temperatures. And if you don't have the drug, that protein will denature and aggregate and come out of solution. So you can actually just look at the levels of the protein to determine whether the drug is binding or not. Now, this is not a perfect screen. It doesn't work that way for everything, but it's a good discovery-based approach to take. And you can see that as you raise temperature here, what you find is that when you don't have the drug, you see a reduction in the levels of the protein that remains in the supernate, and it doesn't aggregate. But when you add the drug, you can stabilize it. See that here. So one of our hits I'm showing you is PEP-T1. So PEP-T1 is a dipeptide transporter, so that fit with a lot of our yeast worm data. It was identified in 1994, and it belongs to this specific family of transporters. This one is in the enterocytes of the intestine, so it's involved in uptake of dipeptides and tripeptides. And like most of these receptors, it's not specific for one amino acid. It'll take up any amino acids that are small peptides. Now, you might think that if you read textbooks that the way you take up amino acids in the intestine is you have all these clean, elegant single amino acids and individual transporters that bring them all into the cells. And there are individual transporters for specific amino acids. But research has shown that most of the amino acids you get from the gut are not in that clean, elegant form. They're small peptides, dipeptides, tripeptides, etc. And it's receptors like these that bring them into your body. And I want to point out that one of the main drivers of MTOR activation is amino acid levels. So people talk about insulin IGF signaling, and that certainly can activate mTOR, but also amino acids activate mTOR through this RAG complex. So when you have reduced amino acids, you have reduced mTOR signaling. And we and many others have shown that elevated mTOR signaling later in life is a big driver of age-related complications. And if you inhibit TOR, you can extend lifespan. So one thing that downstream Gemfibrozil might be doing is reducing mTOR signaling. That's actually shown here. So you have TOR in an inactive state. When you add amino acids, you create this complex on the lysosome, which activates mTOR. And so that we wanted to test this with Gemfibrozil. And so you can take cells that are starved for amino acids, and now we're looking at phosphorylation of S6 kinase, which is a substrate of mTOR kinase. What you see is under starved conditions, you have without the drug, you have very low signaling, and the drug doesn't affect that. If you add single amino acids, you activate mTOR, and that's not affected by the drug, as you might expect. If you add just leucine, you can activate mTOR, and that's not affected by the drug. But if you add dipeptides specifically, you activate mTOR, and then the drug blocks that activation. So this is again consistent with this link between Gemfibrozil all the way through mTOR signal. Now, interestingly, we did not pull PPAR alpha out of the screen, and we see no evidence that this drug activates PPAR alpha. I'm skeptical that it's actually a fibrate. I would point out that other fibrates, and there are a bunch of them on the market, do not have the aging phenotype, at least in the yeast and worms, and don't affect amino acid transport. So we think Gemfibrozil is doing something entirely different from this other class of drugs. So we were interested in what else we got out of the screen too. And one of the things we got was FABP4, a fatty acid binding protein that's becoming more and more studied because it has a number of roles in driving atherosclerosis and also driving altered lipid metabolism, diabetes. And so now we are currently working with the model that Gemfibrozil may be inhibiting FABP4, and that may be what's going on with regard to the hyperlipidemia. So I'm skeptical of PPAR alpha at all. I think there are two targets we've identified, FABP4 and PEPT1. PEPT1 links to aging, FABP4 links to hyperlipidemia, and probably it may also link to aging. There may be connections there as well. So there may also be other targets, but we're working very heavily on these two. We're doing mouse aging studies right now with Gemfibrozil. It looks like we can reduce frailty in the mice, although that's still preliminary. And if that works, we're going to be thinking about how we can do human intervention studies. So I want to switch over now to work with Ponce de Leon Health, looking at natural products that have effect on lifespan and healthspan. This first mouse data was published in 2020, so I'll go through it really quickly. But what we were trying to do is find individual natural products that have effects on aging using a mouse model starting at 18 months of age, looking at survival in the mice, but also looking at a whole range of age-related phenotypes, most notably frailty, which is this Susan Howlett's frailty index, which is 31 phenotypes, which are non-invasively analyzed during mouse aging. And so this is the key piece of data. Or sorry, the next slide is the key piece of data. This is showing that AKG affects lifespan in the mice. It's a modest effect. It's about 5 to 10% in females and significant, closer to 5% in males. And if you look at one survival curve, it's not significant. We've done this 4 or 5 times and we get similar results. So there is a modest lifespan extension in the mice, but the key thing is that there's a dramatic frailty reduction. And so this is the frailty index. The higher the score, the more frail the animal. The frailty goes up faster in the control than the AKG treated mice. This is a little bit confusing data because these mice are dying along these months of analysis. So we went back after the mice died and calculated what percentage of the mouse's lifespan they were at when we took the frailty measure, 50 to 60, 60 to 70, etc. And what you see is that frailty goes up steadily in the control animals and suppressed by about 50% in the AKG treated animals. This is females, it's also true in males. And so we think we're compressing morbidity with this molecule, big effects on health span, modest effects on lifespan. So what is alpha-ketoglutarate? If you've taken biochemistry, you should know this is a central TCA cycle component. In fact, it may be the rate-limiting component of the TCA cycle, which is involved in energy production and shuttling around different metabolites in the cell. AKG particularly is at this interface where amino acid breaks down comes in to generate AKG. So you can have crosstalk between carbohydrates and amino acids through this molecule. It goes down with aging, and supplementing it back up seems to be beneficial. What we've shown is that we can improve adult stem cell function, better metabolism, better respiration, cool effects on the microbiome, which we're just waiting for before I can see what's going on. Pretty dramatic reduction in inflammation, and I'll show you some human data on biomarkers. The problem with AKG, and this applies to NAD and any of these other natural products that are central metabolites, is that it does a lot of things. It's involved in about 800 different reactions in the cell. So trying to figure out what it does is a challenge. And we spend a lot of time and effort now trying to AKGs affecting aging. It's involved not only in the TCA cycle, but it activates or regulates these dioxygenases, which regulates prolyl hydroxylases, which affect HIF1 alpha, which is a known longevity molecule. It has clear effects on immune function through glutamate. It also is a substrate for DNA demethylases, so it could be affecting methylation clock and epigenetics that way. And trying to sort all this out is an interesting challenge. And so in humans, what we chose to do is to use these biologic aging clocks. We chose for the first study we did was based in just people using the PDL product. And so in that case, we used a very simple biologic aging clock called TrueAge, which looks at a number of methylation sites and tries to estimate your biologic age. And so this was the first study we published. It's actually it's out in Aging now. 42 participants. This is not a placebo-controlled study. I'm telling you up front. We couldn't do it that way because at the time we did this study, we could only use people that were taking the product. So they took it an average of 7 months. 14 females, 28 males, average age 40s to 60s. And we measured this TrueMe test to see what the biologic age was. And so this was the data. So we're looking at chronologic age minus the biologic age here. So things that go up means the biologic age is going down in 7 months. And you can see that most people got biologically younger, at least by this test, in this period of time, some dramatic effects. And I wanted to say right now that I do think there's probably a bit of a placebo effect to anyone who believes they're taking something to slow aging. That may be a couple years. That's sort of what I believe based on a whole range of studies that have been done. But this is a 7-year effect. And so if this is true, this is a pretty dramatic effect on biologic aging. And what makes me interested in it particularly is that we saw interesting trends here. So we saw there were 2 factors that predicted your response to the product. And I should say that this product is not just AKG. It's a combination of AKG and low-dose vitamin D for females and AKG and low-dose vitamin A for males. It's called Rejuvenate. What we found is that if your baseline chronologic age was higher, you responded better. And also, if your biologic age, the older it was compared to your chronologic age, the better you responded. So basically, it was the people that were not aging particularly well, at least by this measure, that responded the best to this intervention. Now, I don't know if that's going to be true of all aging interventions. After we show this data to people, they're saying, yeah, that's what I've expected that all along. But I have to say, I didn't expect that. So it'll be interesting to see what other interventions do compared to this going forward. We've now completed a clinically controlled study, placebo-controlled, 100 subjects, which is pretty big for a natural product company. And we're analyzing the data right now. There's some interesting things happening, but we're just trying to make sure we have everything clean before we talk about the data too much. So I just want to close by talking about some of the stuff we're doing on the academic side in US. We now have our Center for Healthy Longevity. up and running, active studies going on. It's very feng shui. You should come visit if you're in Singapore. We'll take a bunch of measures. And so clinical studies are finally up and running, I think, at a pretty fast rate. A lot of that credit goes to Andrea Meyer, who's a biogerontologist who's come to work with me at NUS now. And so we're partners in crime. And our strategy really is to validate interventions that work in animal models, optimize and begin this process of personalizing so that we know which people are going to respond best to which interventions, and to choose things that we can scale. So both when it comes to measuring aging and interventions, on the academic side at least, we're looking for things that are natural products, repurposed drugs that we can move into the community, things that don't have extremely high cost, and that we can do without taking pieces of people to get answers. So nothing more than a blood draw to measure biomarkers. So I mentioned this study already that's been done by PDL Health at Indiana University that we're analyzing the data. We're going back now and doing another study with just the sustained release calcium EKG at NUS, and we're without the vitamins, and we're adding a whole range of other biologic aging markers. And we're using this as a prototype because the approach we want to take is not specific to just AKG. We want to compare different kinds of interventions in a relatively agnostic way to see which ones work best in which people. So we're taking both healthy people and early pre-disease people, middle age, doing these 6 and 9 month studies, looking at a wide range of biomarkers, different kinds of interventions, and to begin this process of figuring out who responds to what. But the world changed when we got to these omics-based molecular pattern biomarkers like DNA methylation. We have a proteomics clock now that we haven't published yet. There are clocks based on metabolomics, transcriptomics, microbiome, and they're fascinating. I still think none of them are completely validated, but we're using them at least as endpoints to see whether interventions work or not. So we're doing DNA methylation, looking at autoimmune panels, and also some hallmarks and pillars and cell senescence, for instance, blood panels. We have a facial aging clock with Jackie Han in China that we're working on. And then we're coupling those to more standard measures of aging, like pulse wave velocity, other clinical parameters, physical function, etc. All of these different measures, what we'd like to do is to see how they interact with each other and get to a point where we can just do a few of them and scale it to maybe the whole Singapore population when they reach middle age. And we now have DNA methylation data on 10,000 Singaporeans across a wide age range. That should be publishable soon. I can't tell you about anything today, but there's some very interesting features regarding sex and ethnicity and biologic age from these studies. We have other measures like telomeres, things in these people as well, and genetics. And then the human intervention studies are just getting started. AKG will be the first, but we're going to do 5 to 10 of them. And we have a wide range of collaborative studies with other groups in Singapore that I don't have time to talk about. So that's kind of where we are. We have a big healthy longevity program now in the US that has about 35 faculty in different themes. And so there are lots of people working on aging now. We've just started our Asian Center for Reproductive Longevity and Equality. This is targeted toward ovarian aging, both what causes ovarian function to decline and what are the consequences of menopause on long-term health in women. So there's some exciting science going on there. And I think many of you may know, but we have, slide kind of got screwed up, but there's a webinar that we do Thursday nights at 7, where we talk to people in the aging field, scientists, clinicians, investors, etc., and try to bring everybody together to understand features of aging and longevity. So this is free. I don't have the website on here, but if you just search my name and NUS Healthy Longevity, there's a free registration. All this old shows are available on YouTube as well. So happy to close. I just want to make one last point, which is that we really want to work collaboratively. So if you have interventions that you're interested in, both from a private sector side or academic side, or if you have that you want to study in mice or that you want to study in humans or even new biomarkers, we're happy to talk about collaborating on those things and incorporating them into our studies. So you probably know where I live, so you can reach out and we can talk about those things. These are the people in the lab that have helped out, collaborators. And just once again, Andrea has been really helpful in getting the clinical stuff moving more quickly and to bounce ideas off of. So I'm happy to stop and take questions. Thanks for your time.
Thank you, Dr. Kennedy. Yes, we got a number of questions that have just come in, and we've got some time, so we got some time to answer them. So the first question that I have is, and hopefully I got some time for some of my own. Did you test the intervention, the AKG treatment, at different age points? And if so, was there a difference in lifespan extension with animals treated for life versus treated for life versus at a middle age stage, I guess, or at an earlier stage? So was this intervention attempted at different stages?
Yeah, we haven't done that. It's a great question. You know, that we started at 18 months, and that was largely based on the rapamycin data that it has such a robust effect there. But that doesn't mean every intervention is going to work that way. And we should go back now and do earlier stage interventions. What we did choose to do, and we're just completing these studies now, is to do different doses starting at 18 months. And we've changed our studies as well so that we're not doing survival anymore. We're doing 6 to 9 month interventions and using biomarkers and frailty. And the reason for that is that it aligns things more with the human studies, and we're trying to use the mice to understand humans. So we decided to try to line things up more. It's also faster, cheaper studies, so we can do more interventions as well. So we're trying dosing thing first, but I think it would be great to go back and look at earlier and even later interventions to see what happens. And most that question would apply to most interventions in mice too. So.
And I actually got a question related to that, because when you showed the slide about regarding compressed morbidity, and you're comparing the mice data to human data, so when we talk about compressed morbidity for humans and a frailty index, is there a standard metric for a morbidity score? So if we say somebody's morbidity is compressed in humans, what does that mean quantitatively?
Yeah, that's a good question, and there's not really a great answer for it, and I'm probably not the person to even attempt the answer. But you know, even with frailty, there's a lot of different ways you measure frailty in humans. So there's like a Fried index, which couples like ability to function in life with a little bit of physical studies. There's like a SPPB, which is more of a function measuring your balance and muscle strength. So there's a lot of different ways to do frailty in humans. And I think that would apply for how you would define morbidity as well. So it's a bit of a subjective statement. I agree.
Okay. Yeah, I guess Keith has a question here. Does this relate to accumulation of deficits indices? I've heard that. Yeah.
Yeah, that's one way to think about frailty, and that's a more population-based, broader assay. You know, things we'd say that the sort of functional behavioral measures that the humans would be a good way to correlate, that are more like physical strength behavioral things, because it correlates better with the mice. But you know, this is a topic for a whole debate, how you best measure that.
Aaron's got a question. Is there a graph of AKG versus age somewhere that shows the degree of loss over time?
Yeah, this is it's published in an old study, and there's not a good graph in that study. I will say that AKG is a bit like NAD, and that if you look at the bloodstream, it's not much there. And so even if you treat the animals with AKG, the levels go up very fast and down very fast. And so I think that it's a little bit hard to measure doing blood analysis. If you look at tissues, you get a bigger effect, but there's not much of that data in humans. So we're basing our statements on the mouse data and also some published human data, which is quite old and should be repeated.
And somebody had a question here that was kind of pretty direct, which is that there are supplements available on the market. I believe calcium AKG. Is there? Do you have an endorsement for taking those, or?
I'm biased because I work with PDL, so I'll just admit that up front. We believe that the sustained release version is better, basically for the same reason I just told you. it doesn't last very long. Even the bacteria in your gut can use it. If you have it being released slowly, you get a longer distribution in the intestine, and we think that has a bigger impact. So I would argue for that. But obviously, there are a lot of different AKG forms on the market that people can buy. And we recommend 1 gram a day, but honestly, the dosing hasn't been done in humans either. And I have people get back to the company and say, "Oh, I'm taking 6 grams a day, and my hair is growing back, and blah blah blah." And so we should do some dosing optimization in human studies as well. It's just a matter of resources. And by the way, that statement was anecdotal, and so I'm not saying that to take 6 grams a day. So.
Gotcha. So another question is that in the past, you talked about testing combinations of supplements and finding that some combinations prove to be detrimental. Could you give some examples of detrimental combinations?
Yeah, so we find that metformin, at least in mice, well, not metformin, we found that, what's the name of the compound? There's a natural product that's in Ayurveda that's has very similar effects to metformin, and I'm losing my mind. That one is, when we add it with AKG, it cancels each other out. Honestly, we don't see beneficial effects of NAD precursors in AKG either. But I must be still not doing things perfectly, because when we use NAD precursors, we see very, very, very small effects in mice. But when you add it with AKG, it cancels out the AKG effect. So the point I would make is that, especially for people out there that are doing a lot of supplementation on themselves, is that when you start combining things together, you get unpredictable outcomes. And so if you're taking 12 things, I have no idea what you're doing. So it's, I tell people to keep it simple, at least for now, because we just don't understand how these combinations work together.
Gotcha. Now, these studies probably haven't been done. So I just thought of a follow-up question, which is, when you're talking about taking these things in combination, we're assuming you're just going to eat them or take them somehow all at once. Now, if people look into staggering taking supplements that are not at the same time, but taking one supplement one day, another day, is there, can you have a time differential? Would that make matter?
Or even like taking it for 3 months and switching to another one for 3 months. Yeah, that I think if you put a gun to my head and said I had to take a bunch of different things, I would take some kind of strategy like that. But it's all speculation. Experiments haven't been done. A lot of it's also based on rapamycin, which, at least in mice, you can give for short time windows and have effects for a long period of time. So the thought process is that if you can do that with other interventions, that maybe you can get the benefits of one and then switch to the other and then switch back. I think that what we don't know is whether other interventions will have that long-lasting effect, and whether even rapamycin has that effect in humans. Humans may respond differently than mice in terms of the long-term effects of the drug. So all interesting questions. If we had more resources, the field could answer these things, but the mouse experiments are still really expensive, and so it's you have to prioritize what questions you're asking.
Right. Okay. We got a couple more questions here. So with the lifespan and C. elegans studies, how much is the lifespan of C. elegans extended after Gemfibrozil exposure?
It's not huge. It's about 30%, if I recall, 25 to 30%. So it's analogous to a lot of other drugs, but of course, as people know, with genetic interventions, you can get huge effects in worms. That largely has the effects of drugs are smaller, and that could be simple problems of getting the drug into the worms. You have to, put the drug in the media or put it in the bacteria, and the delivery with drugs is not perfect for worms. So that could be some of the effects.
Another related question to the Gemfibrozil studies. Do you think the same patterns would emerge if less MTOR activating amino acids had been utilized, such as cysteine?
Good question. It's really hard to dramatically change your amino acid composition in your diet. So, I think that in terms of an intervention that might work in mice and humans, it's probably has, will affect most people if it works through this mechanism. But we haven't gone back and tried to look at, well, we're doing a little bit of this, looking at specific dipeptides to activate mTOR and seeing whether Gemfibrozil affects that or not. And whether if you use specific dipeptides, for instance, in a yeast that doesn't activate mTOR as much, whether Gemfibrozil would affect lifespan or not. So those are good questions, you think, things that could be done too.
We'll get time for just one more question and then we have to wrap it up. You had a slide where you showed for human studies a alteration in biologic age using a, I believe, a methylation clock, and you said something where you said that since you didn't have certain controls in place, the placebo effect is something that we always want to look out for. How much of a placebo effect would there be for certain biochemical markers such as methylation status? And is there a general placebo scale for certain
there isn't,
metrics?
but I just believe that if you're paying money to be younger, you're probably a little bit younger. I don't think it's a big effect. I think it's probably a relatively small effect, maybe a couple of years, but that seems to be something that's being witnessed in other studies. And also, it may be that if you suddenly become conscious of aging and you go buy a product like Rejuvenate, you also decide to eat a little bit healthier and exercise a little bit more. And so I'm just flagging that, saying that, the placebo control, I want to make sure everybody knew that, and also that we should try to do the controlled studies whenever possible, because I think for something, methylation is alterable. All of these biomarkers seem to be alterable. So we need to be conscious that we're actually having things that are in the pills having the effect and not just paying money for the product. So